Chris Armstrong. I'm the Acting Chief Scientist and Engineer. I'd like to begin by acknowledging the traditional owners of the land, the Gadigal people of the Eora Nation, and pay my respects to their elders past, present and future. For this morning's breakfast seminar, we welcome one of our most eminent researchers, Professor Justin Gooding. Justin is from the School of Chemistry at UNSW Sydney and is co-director of the New South Wales Smart Sensing Network, an initiative that enables researchers to push the boundaries of smart sensor technologies, bringing together experts from different disciplines to address real-world problems for industry and government. He's an ARC Laureate Fellow, also co-director for the Australian Centre for NanoMedicine at UNSW. This also draws together expertise from UNSW's faculties of engineering, medicine and science, aiming to find solutions for hard-to-treat diseases. Justin leads a very strong team of 45 who investigate surface modification and nanotechnology as they relate to biosensors, biomaterials, electron transfer and medical applications. His current work includes development of plasmonic sensors, which can be used to detect bacteria such as salmonella and may help in the diagnosis of viral infections, cancer and other diseases. His application of the use of light-activated surfaces to the detection and understanding of circulating tumour cells is enabling analysis of the genetic makeup of cancer cells and the testing of new drug therapies to treat existing tumours and prevent their spread. Equally, his team's expertise in nanoparticles is helping to enhance tumour contrast in MRI and other imaging techniques, as well as being used as theranostics. The theranostics are materials that can diagnose diseased tissue at the same time as delivering a therapeutic treatment. Justin's research was recognised by the University of New South Wales through the awarding of its highest appointment, a Scientia professor in 2011. He was elected a Fellow of the Australian Academy of Science in 2016, and as I mentioned, he's an ARC Laureate Fellow. Other highlights from Justin's many awards. I printed out his CV yesterday and I thought I printed triple, but it's got a very long CV and has many awards to his name. The New South Wales Tall Poppy Award in 2004, a Royal Australian Chemical Institute Lloyd Smythe Medal for Analytical Chemistry in 2007, an H.G. Smith Medal for Chemistry from the RACI in 2011, a New South Wales Science and Engineering Award in 2013, a Faraday Medal of the Royal Society for Chemistry in 2016, 2017 Australian Museum Eureka Prize, which was his second, for an outstanding mentor of young researchers. Join me in welcoming Justin as he explains how sensors are changing our world.
Honourable members, distinguished guests, ladies and gentlemen, firstly let me thank the Acting Chief Scientist for the nice, somewhat long introduction and also the just the invitation to come here. And I'd like to thank you all for sacrificing your early mornings and come to hear me speak. When I first heard of these breakfast seminars, I had this vision of me boring you all to tears and some of you waking up with your scrambled eggs on your face. I'm hoping that doesn't happen. So what I'm going to talk to you about today are three things. As you can see, I've got many hats, which is common with academics these days, but I'm going to talk to you about three things related to sensing. I'm going to talk to you about maybe the big picture of sensing, you know, not just the sort of sensing I do, but how sensing is really impacting our lives. I'm then going to scale down a little bit and talk to you about the New South Wales Smart Sensing Network and how that's helping us here in New South Wales address some of our needs where sensing can actually provide a solution. And then I'll scale down to the own work that my own research group does on ultra-sensitive sensors for biomedical applications. But before I get to the science, I wanted to acknowledge the people that give us the money. They're the dominant people that give us money. I wanted to acknowledge the group as a whole. Many of them are here. The group is now co-led between myself and Professor Richard Tilley, and these are some of the long-standing collaborators that I'd always like to acknowledge because some of them have worked with me for many years and allowed us to make the sorts of progress that we've made. So what unifies this big group of people is one thing, and we're chemists. And what unifies us is modifying surfaces with a high degree of control. We modify surfaces by self-assembly using single molecule thick films. And why we do that is because then we can change any sort of material and give it completely new character and completely new functionality. And we exploit the fact that, as represented here by the the red and the blue, atoms at the end, we can put multiple different components in and get a molecular level control over how that surface now functions, how it interacts with its environment. And so having a surface that interacts with an environment in a highly controlled way is the backbone of chemical sensors. And so using that sort of idea, we then work in many different areas, and mostly related to making surfaces interact with biological systems and measure things. And so we're really focusing on, and this is going to be really important, making surfaces that measure things, but not just measure but the same idea can be exploited for measuring many different things. And as I said, that's going to be important. But what I'm going to talk to you about today is a little bit about sensors, and then progress towards ultra-sensitive sensors, moving towards single molecule sensors, and how that's really going to change the landscape in chemical sensing. But sensing as a whole is much bigger than just chemical sensing. And so I thought it was at least prudent to say what we think a sensor is, because I have to say that when I talk about sensors, and what I mean by a sensor is often very different to what other people mean. And so a sensor, we could say, is just a device, a module, or a subsystem which detects an event or a change in environment. But that actually just sounds like almost any analytical method, any measurement tool. The key thing here is in the environment, and the key principle is we go away from finding me in a lab, a very rare event, and do the measurement out in the field where we need that information. And I think that's the key concept of a sensor, is that it's done where you need the measurement in the field. And so that's the sort of definition we'll take with us. But from that definition, really, sensing has completely pervaded our world. And you can classify sensors in two very broad ways. You can talk about physical sensors. These are sensors that monitor things like temperature, pressure, accelerometers, gyroscopes. They're measuring physical changes, and they're ubiquitous and everywhere. But along that line, most physical sensors are non-contact sensors. They don't have an interface that interacts with the thing you want to measure. They just measure it somehow. Pressure, temperature, for example. The other type of sensor is a chemical sensor, and these are where we actually have to measure a specific chemical species, not a physical phenomenon. And not exclusively, but most frequently, they're interfacial devices. And that's where our skills as surface chemists come in. We make materials that we make a layer of molecules on that surface and put molecules that are absolutely selective for the thing we want to detect, so that our sensor only responds to the thing we want to detect. Now, of these two types of sensors, they're very different in their development and how ubiquitous they are in our lives. And a classic example of how ubiquitous physical sensors are in our lives comes from our car. So if you look at a modern car, I think many of us in the audience will remember when a car, you lifted up the boot and there was an engine and something went wrong with the car and somebody turned the keys and they looked and listened, and the only real sensor you had was the amount of fuel in the car. Right? You at least had a dial that told you the temperature and the amount of fuel. A modern car has, even in the engine, it has sensors for all sorts of different things. Hundreds and hundreds of different things, and then there's sensors all on the outside. So really, what that's done is give us a car that, well, breaks down a lot less, though we may not think it, it's actually true. It breaks down much less. We get those really annoying lights that tell you, take it to the mechanic because it's about to break down, but that car is also more fuel efficient. It runs better. It doesn't break down as often. So those sensors have created diagnostics such that your car now is a much better car than it ever was before, though could be a little bit annoying. And basically a modern car has about 20% of its values in the electronics, has between 70 and 150 sensors in it. About the only sensor this car doesn't have is a sensor for a giant lobster falling on its roof. And actually I thought about that today, actually, when I walked in here. I really wished I had a sensor that detected things falling, because I got hit by a bird doing its morning constitutional. And I thought, I'm going to have splotches all over me, and I did, and I thought, I've got to clean them off. off. Otherwise, you're going to talk about to your friend. Do you remember that time we went to that talk? And you know the guy that got by the bird? What did he talk about? Got no idea. So, but basically, these physical sensors are everywhere in our lives. Obviously, we carry them around with us everywhere. This is full of sensors. My watch also has got about 8 sensors in it, telling me all sorts of things. So they're ubiquitous in our lives. And so it's really big business. And when I say big business, I mean annual sensing market's about 270 billion globally, escalating by 2021 to about at least 400 billion, probably more. And that really is driven by, and this is almost entirely physical sensors. So it's about 23% of the market is sensors in vehicles. Another 23% is the IT industry, so that's your cars and your phones. Then you can think there's about 18% is, or 19% is industries, about 18% is building, about 3% is at home, and that's obviously escalating enormously, and about 9% medical. And of all those things, the 9% medical is really the chemical sensing. All the rest is predominantly physical sensing. And it's really driven, and that has really driven the idea of the Internet of Things. And so by 2020, we think we're going to have about 4 billion people connected, and there's going to be what's 25 billion embedded intelligent systems. That's 25 billion sensors floating around the world, giving us information about everything around us. And that then creates a huge problem, 50 trillion gigabits of data. So with the Internet of Things, what are the drivers? Because that will tell you, that will really lead you to why I want to talk more about chemical sensors. So here's the main drivers for the Internet of Things. Things like latency or data delay, so that's if you've got your virtual reality headset on and you move your head, the software moves straight away. It's data rates, processing the information, storing the data. Ease of integration might sound like it's a sensor-related issue. That's actually getting the sensor to do more processing before it sends the information, so less information is sent, and a little bit to do with power, so the sensors use less power. But the key concept of all these sensors is they're really going to sit there remotely in the environment and measure things that we need measured and send back information. And so nearly all the drivers are related to the data and the electronics. There's some drivers in low cost, because the lower cost the sensors, the lower the power, more that we can have distributed, the higher the fidelity of information. But really it's nothing to do with the sensor. There's not much to do with the sensor. And so about 10, 15 years ago, the big players in data, because it's all about data, a big computing company started to distribute software related to making sensor arrays and addressing sensor arrays. And it was all really driven by this fact that there's going to be, people perceive there's going to be sensors everywhere. Now, Hewlett and Packard actually said, they came up with this idea of the central nervous system for the Earth. This is called SENSE, very clever. Which is this huge array of sensors, 1 trillion sensors sitting there remotely in the environment, measuring all sorts of different things. This is the vision. So these guys are all interested in the driving computing power, but this is a different vision again, because one of the phrases said was, we're going to use these sensors to monitor things, anything from electronics and tracking hospital equipment. That's sort of what we have, that's quite common. But then it goes from sniffing pesticides and pathogens in food. So it's progressing from measuring the physical changes to the chemical changes, just measuring chemical species. And that is where the unmet need is. It's the chemical sensors. So the unmet need is that if you want to have a sensor that sits remotely in the environment and measures everything, whatever it is that you want it to measure, it needs to keep doing it, and needs to be able to do it repeatedly over and over and over again. And the physical sensors are very successful at doing that, such that really, as you saw with the Internet of Things, the issues are not so much related to the sensors. The sensors can be improved, but the big shifts are not going to be in the sensors. There are some examples of chemical sensors that we can do that. We can just leave them remotely in the environment and they can measure things. pH is actually one example. Gas sensors, these are the Figaro gas sensors. These can be done. How they work, it was first developed in 1969, so it's almost 50-year-old technology. They have a metal oxide and they just measure resistance. And when gas is absorbed, the resistance changes. Why they can keep being used is because they do it at elevated temperatures so they can drive the gases off. And so they can be reused repeatedly. And spectroscopic sensors, where they don't actually contact the sample, they can also be used repeatedly. But most other chemical sensors can't, and that's the weakness in that Hewlett and Packard vision, is we don't have the sensors that can measure chemical information that can sit out in the environment indefinitely, or we don't have many of them. And so what's the problem? What is the challenge? And the challenge all comes down to the fact that they're interfacial. They're basically a surface that contacts with the thing you want to detect in a well-defined way. So this is my cartoon. So we want this type of cell to bind to the surface, not the red blood cells or not these bacteria. I have to say, when this cartoon was drawn, I think the person didn't think it through. So, you know, you're going to detect your healthy mammalian cells, and if you see how many bacteria are in there, that is a very sick person. But beside that, so basically what we do is we modify a surface with chemistry that will selectively bind to one thing and not the other, not everything else. And of course, the one thing we want to detect is the rare thing most frequently. It's not the common thing. And what we want that surface to do is stop all these other things binding on it. And they're way more prolific on the sensor, or in the environment, the sample that the sensor is measuring. And so though we can make these surfaces pretty well, and I'll actually show you some later, we can't, they're not perfect. And so if there's 1 in a million molecules as a molecule of interest, an occasional molecule that's not meant to bind to the surface does, eventually that surface fouls. And so the challenge is not making a sensor that works once, the challenge is making sensors that work over and over and over and over again. And so that's been a big problem. But it's not as big a problem as it could be, because we can make surrogates. And so the water industry is a classic example where we use surrogate sensors to give chemical information. So obviously, if you want to monitor your water, you need sensors that are sitting out there remotely being used all the time. So they measure things like turbidity, so that's, you know, whether there's particles are there, conductivity, so how much salt, temperature, dissolved oxygen, pH. They measure things like that, and they can make remote sensors that can do that. Because they have some a priori knowledge about what's upstream, where the water comes from, then huge amounts of chemically related information can be used by measuring things that are surrogates for the chemical change. So it's a very effective strategy, and a strategy that I would say is the strategy to take whenever you can. My watch does the same thing, right? It's got 3 little green diodes that say where my heart rate is by looking at the flow of the blood. Some of these watches have skin conductivity sensors to see whether I'm sweating. And so they're really giving surrogate bits of information, non-chemical information, to say what my health state is like. And I can tell you, when I'm running, it's not as good as I'd like. But we do need methods that, if we want to detect specific organic chemical species or specific biological species, we need sensors that can do that. And that's a weakness. And that's really where the unmet need is in the sensing field, or one of the major unmet needs. That's where we're going to get a big shift in capability. And so you might then say, so that's all very interesting, so what's New South Wales doing? So if we look at the Premier's priorities, there's a whole lot of priorities on the website, and if the ones I've marked in red, creating jobs, delivering infrastructure, improving surface levels in hospitals, keeping the environment clean, tackling childhood obesity, they all have sensing-related aspects that can contribute. And so as a result, the New South Wales government, the Chief Scientist and Engineer, former Chief Scientist and Engineer Mary O'Kane, talked to Ben Eggleton from the University of Sydney and myself, and talked about, well, what can what New South Wales do? And I have to say, you know, huge raps for Mary. Ben and I had never met each other. We knew each other. We knew of each other, but we'd never met each other, but she clearly worked out that we're very complementary in the way we do things. And so about August 2016, we launched what's called the New South Wales Smart Sensing Network. And so it's really designed to bring pragmatic solutions to problems that the state faces, or challenges the state faces, chemical or measurement challenges the state faces, and basically connect the state government or industry to the university. So it's really meant to be a portal that allows, if you've got a sensing challenge or a challenge that might have a sensing solution, you can come to the Smart Sensing Network. We can talk through the project, what the needs are, and if you've got some money, you can then fund some work, and we can connect you with the right the right people. And so in the initial phase, the right university researchers across the state. So in the initial phase, we had a series of pilot projects really just to show that there is need and there is the capabilities in New South Wales and to attract people to the network. And so we had wildlife projects, actually a very interesting project. They had a project on tracking koalas using cameras. And you'd think, well, how do you decide, identify one koala after the other? Is it the fluffy ears? Is it the nose? It's the backside. Koalas have a characteristic backside. We had projects on wearables, projects on water quality, projects on air quality, and projects on biomedical. And so then in February 2017, we had the launch. You can see the two vice-chancellors from the two different universities, Mary O'Kane, us two at the back, and the chair of the board, Susan Pond, who's here, Professor Susan Pond, who's been a huge driver in this as well. Really, I don't think you could dream about a better chair of the board, really helped us shape the vision. And so then we started developing some products or some sensors to address issues that were prevalent in New South Wales at the time to sort of show the capability of the network. And so the network is actually morphed from that initial phase now to being a portal. So we have the network, which is really the Department of Industry money that's used to fund the network, funds the central hub, the office, where there's people that then connect the member universities. We now have 6 member universities that contribute to the network, and there hopefully we can form the connection between the government, industry, and the right researchers to solve the problem. And so we use this mantra of there's a local challenge, something that's important for New South Wales, that's got a global solution. And so that if we can develop the sensor for it, that sensor then can become commercial. And so not only have we solved the problem, we've generated revenue base and, of course, jobs for people in New South Wales. That's the vision. And so we have the 2 co-directors here who are sitting at the top with a members committee, so that's all the different university representatives. There's a board which has just been reappointed, and we've just appointed a new chief operating officer who's hopefully going to do most of the work. And we have business development people. We have a fantastic project administrator and Inca, and then technical theme leaders. And so these people are mostly here in the room, so if you're interested or if you've got issues that you want to talk about, then try and track them down or track me and I'll point you to them. And so we have the theme leaders have expertise in different themes related to areas that are important in not only our state but everywhere else. So I thought I'd show you just some of the projects that are done through the network before I get to more of the work that our group does. And so, and I want you to really get across this idea that the network is not about academic researchers doing their stuff and hoping someone finds it useful. It's really about the end user driving what they need and us coming up with the simplest pragmatic solution possible. And that may just be using off-the-shelf components and repurposing them. And if that can be done, that will be done, or it might be developing new technologies. And so Ben has a project in University of Sydney with Tom Honori, who's actually here, who's the main driver, where they take cheap off-the-shelf electronic components to make these very cheap sensors that measure particulates. The driver at the time was when we started was the issue with this issue of dust from coal trains, but of course it has, like everything we do, it shouldn't be just applicable to one area. If you think about all the infrastructure, the tunnels that we're building, sensors that can measure particulates, a fantastic surrogate for a lot of things in the air. And so they basically make these cheap sensors, they go out and distribute them near places, and the idea, if they're cheap enough, we can get very high fidelity information, essentially get a map of the dust and how far it goes or where it's penetrating. The key point here is that all the innovation's really in taking the data and converting into information. Another project, but one of the things we learned when we first started is many of the projects, I didn't say one thing about it. I did mention it briefly, but I want to reiterate this point. A sensor, a chemical sensor, is traditionally designed so that you have a surface and something on that surface that selectively binds to the thing you want, with nothing else binding. Implicit in that statement, of course, is that you can't detect anything else. You have to have a sensor perfectly designed for the one thing you want to detect. What we learnt early on was that many of the applications, we didn't know what they were going to be. And so we needed a sensing type technology, something that could measure in the environment that could detect just about anything. And so Alex Donald, who's at UNSW, one of the rising stars of chemistry at UNSW, one of the rising stars of science, if you ask me, he's a mass spectrometrist. And so many of you know mass spectrometry just measures how much things weigh so you can identify them. And many of you have experienced an ion mobility mass spectrometer when you go to the airport. That little white slab that they're swabbing on you actually does detect something. It can detect nitrate explosives and some drugs. That's pretty much what it detects. But it's this sort of size. So Alex has redesigned it so it can be that sort of size, little fingerprint type device. And the beauty or the innovation really comes in how he designs these electrodes that turn the material that you've collected into ions so you can measure them. And the other innovation he's done, which is he's done with our group, is he's made these little needles that are modified with surface chemistry so we can pre-concentrate the thing we want to detect. And so because at that time, the per and polyfluoroalkyl substances, the PFOS substances from Williamtown, the fire retardants, it's a big issue in the Hawkesbury, as you know. So that was something that was important in the States. So he tested it on the idea on these things and on these sorts of compounds. And he showed that he'd get improved sensitivity 200 times, 260 times better separation ability. That's the ability to differentiate a PFOS from another thing. That is the weakness of these sorts of machines, is they can't detect that many different things because of the overlap of peaks. An analysis time of 2 minutes compared to the classic with no sample preparation, compared with 30 minutes measurement time plus sample preparation. So very successful initial foray in this area. And so this sort of technology is not limited to these sorts of devices, to these sorts of molecules. And so in principle, if this progresses through to commercialisation, we can detect almost anything you like, and then we change how we make those needles so that we can detect those things. But what it was really designed to is the need in the state is not necessarily to detect. Sometimes we don't know where the challenge is going to come, so we need a technology that can be a sensing-like technology, portable, taken out into the field, and can measure what we need to measure. Another area, so this is one of our own projects, is actually obviously a sensor that prevents something happening is way better than a sensor that detects that something has happened, right? Obviously we need sensors to detect something has happened, but better to stop it. And so obviously, as you all know, being Australians, sun exposure, skin cancer is a big problem. And so one of the things that we all know is that we need to put more sunscreen on, and we need to get out of the sun. And we all know that, but we don't really know what that means. And so we thought, wouldn't it be great if you could have a sensor that could do that? And there are quite a few, it's quite a popular topic now, but our group, actually this person here, Parissa, who's not the model, is actually the scientist, and she's got one of these things on her arm. It's really just a smart piece of paper. And we took an old-fashioned inkjet printer and made a smart ink, which is titanium dioxide nanoparticles, used in sunscreen already. It absorbs UV light. And when it absorbs UV light, it can actually destroy organic matter. So we have a food dye that you would normally eat and a binder that's approved for human use. And then we print it on a piece of paper. And if it's exposed to the sun, the dye fades. And actually now we've engineered it so when this dye fades, it becomes pink, like sunburn. And so this is just a very simple idea, but it's very pragmatic and it's very effective. And it's so effective that actually there's now the New South Wales Smart Sensing Network has produced its first company, Luminosity Innovations, which is trying to commercialise this technology. So that's really it about the NSSN. So now I really want to go to my own work and the own work of the research group as a whole. And that work is badged through the Australian Centre for NanoMedicine. The philosophy of the Australian Centre for NanoMedicine in some ways very similar to the NSSN, the Smart Sensing Network. It's about interdisciplinary research and reaching out to the end users to drive how the research is going to go. And so I co-direct this with Maria Cavallaris in medicine at UNSW and Cyril Boyer in engineering. So it's really interdisciplinary. And what we do is we work in three main areas. Drug delivery. So how do we make nanoparticles that take drugs to where you need them in a more effective way so that you can reduce side effects? Imaging and diagnostic technologies, which is mostly what I work in, but we do some work in the drug delivery. And then we have social scientists and artists that work on more the social impact of the technology and how the technologies might be adopted. That's John McGee in Arts and Social Science, and Arts in UNSW, and Matthew Kearns in Arts and Social Science. And so I'm going to talk to you about a class of sensor called a biosensor. And that's really where what our group is known for and works. And so it measures a biological species or a chemical species, and the key thing is to do that. Remember I told you that a sensor has a molecule that's really selective for the thing you want to measure? We use a biological molecule to give that selectivity. The thinking being, of course, that we all know that in our own bodies, all our biological molecules are interacting with each other in really, really controlled ways, and only every now and then is there an aberrant event that happens. And so biology is really good at binding to what it wants to bind to. And so here is an example or a cartoon of a sensor that uses nucleic acids or DNA to be the recognition species. And so you have single strands on a surface, and then if the complementary strand comes along and binds to form the double helix, we get a current. But the key point is that we're using biological molecules to give the sensor its selectivity for what we want to detect. So is this really just a fanciful academic idea, or is it worth doing? Well, many of you will know the glucose meters. The glucose meters are the classic example of this. So this is one by a company called Agamatrix. There's many companies that do this. I choose this company because the first PhD student I advised when I was still a postdoc is a technological originator of this company, where he took the ideas from his PhD that we designed together and then turned into a commercial enterprise. And it's interesting with this modern age of data, and I should have said earlier on, you think about how important data is in sensing, as I've sort of indicated, so that's why, the budget actually had 25 million for artificial intelligence and machine learning, or big data, essentially, because that's really important in so many areas. So the idea was, so what Sridhar did was then turn his eclectic PhD idea into a useful company that now makes a lot of money, actually. And so the way what's the biological molecule? So to detect glucose, we'll use the enzyme glucose oxidase that reacts with glucose, and the amount of glucose determines the current we see, which gives us the magnitude of the glucose in your blood. Now, so is this important? Well, obviously, if you know anybody that's used glucose meters, anybody that's diabetic, this is in fact the most successful analytical device in history. There's 10 billion glucose assays done a year. There's many companies that do it, of course. Here's an older one that was about 15 years ago, was on the market. But yeah, this is really important because it turned the lives of people that have diabetes, allowed them to lead a life such that we don't even know unless we see them pull out the instrument. So for many, it's an absolute revelation. And so the latest, and this Agamatrix is important for another reason. This is one of their later devices. It was sold to Sanofi, and it's called IBG Star, and it plugged into an iPhone. So this is actually the very first biomedical device to interact with an iPhone. In fact, Agamatrix, Sridhar's company, was the first company in the world, a diabetes company, to have an app for diabetes management. And it was Sridhar and his partner, Sunny, who went to Steve Jobs and convinced Steve Jobs to go into biomedical sector, which may have some influence on the Apple Watch, I don't know. But basically, it's a very significant company. In fact, it was also the first biomedical device to be sold in Apple stores in the United States, or anywhere, actually. So, big deal. But there's more than that. You may have used other biosensors. You've probably, I guess nearly everyone else everyone in the room has seen somebody use this biosensor where you have a biological molecule, glucose oxidase, telling you how much their glucose is based on a current. But there's others that you may have used. You may have seen, this is an older version, the pregnancy test kits. These are the other big class of biosensor, or one of the other big classes of biosensor. So the glucose meters we call them catalytic biosensors, but they use enzymes, which are nature's catalysts. Pregnancy test kits are what we call an affinity biosensor, because it uses an antibody, so the body's natural immune system, to detect whether a protein's there. And so all you have to do is, like in the glucose meter, all you've got to do is prick your finger and put a droplet of blood. All you've got to do is expose this swab to urine, And you've got antibodies in this window here, this little blue window, that tells you whether you have much of this HCG, which is a hormone that tells you where a woman is in her ovulation cycle, but also it's upregulated when pregnant. And so if you get two blue lines, one is just a control to show the test is working, one is to show you're pregnant. If you get two blue lines, depending on your perspective, you're happy or sad. And so how does this work? So on that strip there, which is the result window, the square one, because I can see I've got that's the coffee. You basically, when you wick it, in the urine is this hormone, human chorionic gonadotrophin, or HCG. And it binds to this antibody, it gets captured. And antibodies just bind to things, that's what they do. There's no effector function, typically. And then in the body is another antibody that comes along, which is labelled with the blue bead. And so this is there, we then form this sandwich structure, and we get a blue bead there, which means we get the blue line. That's how it works. And so modern ones, of course, we can no longer trust blue lines. We have to have something that says you are pregnant. First it went from the blue lines to plus minus, and then of course plus minus confused people, and then it went to you are pregnant. It's actually quite interesting as how you make a device that's to be used by the general public, how many different ways people are going to use it wrong. So it's astonishing. So in the early versions, in the guide it said, you've got to expose this to urine, do not place in toilet. Because some people are, I've got to expose it to urine, they pee in the toilet, they stick the thing in, 9 months later they get a rid shock. So I wanted to go back to these two devices because why they're commercially successful is a really big driver on what we do and why we do it that way. And also why there's not more of these devices relates to. So the glucose device, this is obviously something we desperately needed. It's a huge industry, $16 billion a year industry, getting bigger as our waistlines expand. And we really need it. We do not need this. We have many ways to know whether you're pregnant or not. This is commercially successful for a couple of reasons. One, of course, it allows you to go to the doctor and have a conversation you don't want to have, armed with some information that you don't quite like to know. I think I'm pregnant. You don't want to go, I think I'm pregnant, and you find out you're not. The other reason it's successful is because if you have these antibodies, typically called IgG antibodies, they're structurally the same most of the time. What's different for different things you want to detect is just 17 amino acids in that little cleft or in that side. And that then determines what it selectively binds to. So the same technology can be used for many different, the same basic idea can be used for many different devices. I already said to you before that's one of our guiding principles, and this is why. Because you can get one of these for malaria, you can get the same sort of, this lateral flow device as it's called, you can get it for water quality, you can get it for, in Western Australia, you can get it for drug testing, so you know whether you've got party drugs in your system before you go back up to the mines. So it's used for lots of different things. And so what it's essentially doing is spreading the financial risk of bringing this to market across many different applications. Because to bring this to market costs $100 million on average. In fact, this one only took $20 million, which is really surprising. So $100 million to bring something to market probably means it needs to be $1 billion market. And so that's the challenge with sensing, is not whether they work, or chemical sensing. It's not whether they do whether they work, it's not whether we need them, it's whether we're willing to pay for them. It's a big driver. So where is the opportunities then? Where else could we develop sensors? And one of the opportunities is sensors that work at much lower concentrations. So this works in the millimolar range. These sort of technologies typically aren't quantifiable, but can be made quantifiable. They can work micro, maybe even nanomolar range. So what do we need? So let's look at cancer as an example. So I'm going to talk to you about some of our technologies that address this need in cancer. Though again, they're applicable to many different things. So diagnosis is the best prevention for cancer, or the best cure for cancer. If you're in the early stage, this is for colorectal cancer. If we pick you up in stage 1, you've got 99% survival rate. If we pick you up at stage 4, it's going to be maybe a 26% survival rate. And so the earlier we can diagnose, the more effective is treatment. The earlier we could diagnose means, earlier diagnosis means less amount of material to detect. So how much material are we talking about? So let's say I have a tumour. You don't need to worry, I've got a sensor for it. and it's about 1 millimetre cubed in size. So it's a tiny little tumour. It will excrete about 5,000 of the biomarker protein. I got about 5 litres in blood, a little more, because I had a few coffees this morning. And so basically that means I'm going to have 2 × 10 to the minus 15 molecules of that biomarker floating around in my blood system, which is equivalent to 2 femtomolar. I told you that most of those sensors at best work in the nanomolar range, so that's 10 to the minus 9. So we've got 6 order magnitude to get lower. That is what the challenge is for a lot of diagnostic devices, is if we want to make more that are commercially viable, we need to get down to lower concentrations where we can detect things that we really need to detect at those low concentrations, where the need is there. And so one way of trying to do this, you might think we just need a more sensitive sensor. So if you watch here, so as you can see a little molecule going through this pore, we can detect that molecule. We have technologies that can measure single molecules. So the naive thing to think, the thing most of us think, is, well, all we need is more sensitive detectors. But it's not true. So here's an example, because we can detect single molecules. Here's an example of a nanopore sensor, and here's real data. And you can see each one of these little blips is a single molecule going through a pore. So we can detect a single molecule. To make it a quantifiable sensor, to say how much there is in your stream, we can't detect just one. We need to detect many to get a reliable answer. But you can see what the problem is here. So here's 20 micromolar, not femtomolar, micromolar. That's 10 to the minus 6, so 9 orders of magnitude. And you see we get lots of spikes. Each one of these spikes is a molecule going through and giving a resistance, because we've got an electrode here, an electrode there, and a little hole, and ions are going through there, and if the molecule goes through, it blocks the hole temporarily, and you get a spike, a resistive spike. You can see as we go from 20 micromolar to 8 micromolar, the number of spikes decreases. To 4 micromolar, the number of spikes decreases less. And that's at micromolar. So you can start to see what the problem is. We're going to have each single molecule going through that pore to give us a signal. But we need to be at femtomolar. And at femtomolar, you're waiting about 10 to 12 minutes between each one of those spikes. To do a reliable analysis, you'll probably wait 20 hours, 15 or 20 hours. So the problem is not being able to detect the single molecule. The problem is something different. The problem is that we have our 2 molecules, and we can detect them if they find the pore or the sensor, but they may never find the sensor. So you'll detect them if they find it, but it may never happen. And so we came up with a very simple solution to this. It's pretty obvious. We're not the only ones to come up with it, but there are some differences. We came up with the idea, if the problem is that these can't find the pore because these are randomly diffusing around, and diffusion has time constant diffusion is squared with distance. So it basically means if you double how far something has to diffuse, it takes 4 times as long to get there. We need to reduce the diffusional path length. So our problem is we can detect these, but they can't find the sensor. So we send little nanoparticles out everywhere that are the sensors. That's the key difference to most other technologies. They are the sensors, and they've got antibodies on them that can bind to these proteins, and these nanoparticles are magnetic. So when we capture our thing, we can just bring them rapidly back to the pore or back to the sensing surface in general and measure them. It's not all just about pores. So here's one. So I'm going to show you one example. I'm going to show you a series of sensors that relate to cancer diagnostics in different aspects. And the first one is to detect microRNAs or mRNAs. And what these are short sequences of RNA that the nucleus produces, and they regulate gene expression. They're post-transcriptional regulators. And why they're interesting for diagnostics is that they leave the cell and they float around in the blood in these things called exosomes or other extracellular vesicles. And so RNA is not stable in the blood, but because it's sitting in this little ball, it's quite stable. And so what it means is, for diagnosis, you can access the biomarker directly in the blood. So if you think about the glucose meter and the finger prick, it's now for microRNA. And so all we have to do is, well, all we have to do, it's trivial. All we have to do is extract these exosomes from the blood, break them open so we can detect these RNAs, and we can detect them. The problem is we're down at femtomolar to picomolar, so 10 to the minus 15, 10 to the minus 12. Really difficult challenge. You need to detect several of them, and some get upregulated with a cancer and some get downregulated with a cancer. So they're actual biomarkers for lots of different things, and there's many different microRNAs. But can we develop analytical methods that can detect those? Can we develop sensors? So we took our magnetic nanoparticle idea, and they're slightly different. The magnetic nanoparticles are coated in gold. using our surface chemistry, we modified them with a strand of DNA. Because you know, one strand of DNA can bind to its complementary strand of DNA, just so happens it can also bind to the RNA molecule that has the same the complementary sequence. And on them, they've got a redox-active species. So what that means is something that gives electrochemistry. And so if we distribute these particles through the sample, if they pick up the microRNA, we get a duplex, a double helix, and that changes the electrochemical signal. Now, I'm not going to give you, you are all aware this is a 3-hour lecture, aren't you? I'm not going to bore you with the chemistry, but I'm just going to show you a key result. We can, in blood or serum, we can detect down to 10 attomolar. So that's way below those levels, and that's really unprecedented. And all it is that simple idea of don't make the molecule you want to detect find the sensor, but make the sensor go out and find that. That's just the idea. And so we can look at these particles and compare to the gold standard PCR, polymerase chain reaction, and we can show that if we have a mouse with this lung cancer, we get the same answer, basically. So this works really well, and so this is now starting to attract some interest for maybe taking it towards commercialisation. There's a lot of challenges to overcome. So that's about diagnosis. There are more challenges for diagnostic devices in relation to cancer. I should say this technology, of course, we can apply it to, the same idea can be applied to proteins, all sorts of different things. Like in general, we always try to apply it to many different things. But I'm going to talk about the next stage of cancer and where diagnostic devices could become useful there. So as many of you know, most people who pass from cancer don't pass from the primary tumour. Most pass from secondary tumours, the metastases. And so what actually happens in the cancer cycle is we have the primary tumour. These cells, some cells decide to leave that primary tumour, go into the bloodstream, circulate through and form the metastases somewhere else. This actually, Australian came up with this idea, Thomas Ashworth in 1869. That was his idea that they would have these so-called circulating tumour cells. So the technology we're developing is ways of looking at rare cells and their heterogeneity. So these cells that circulate, these cancer cells, if you can treat them, well then of course you can prevent the secondary cancers. They're very important, not just diagnostic, but prognostic indicators. And so how few cells are there? It's about 1 in a million cells. 1 in a billion cells, about 1 cell per mil. So it's an extreme analytical challenge. How can you detect 1 in a mil? Fortunately, other people have solved that. There's 2 main ways of doing it. There's one way, which is a morphological way, so it relies on the fact that generally cancer cells are bigger than healthy cells. And so then it uses a microfluidic device, which is centrifugal. So the blood is spinning around this device, the heavier cells go out to the walls, the lighter cells stay in the internal wall, the heavier cells to the external wall, so then we can separate. Actually, one of the people who does that, who developed this technology is Majid Wakiani, who works at UTS. And that's been quite effective. Then there's different technologies that can use magnetic nanoparticles with antibodies that selectively, so this is just size. What these ones do is selectively pick up the cancer cells from other cells based on surface markers and the expression of proteins on the surface of the cells. Typical epithelial cell adhesion molecule, or EpCAM. But there's different ones related to different cancers. And so this uses magnetic nanoparticles and captures the cells and then brings them down and does a staining to decide whether they're a white blood cell or a cancer cell. And then people do fluidic devices that use antibodies as well. The key point here, and so this is actually the one FDA-approved device, it's the Veridex cell search system. And this is becoming the definition of what a circulating tumour cell is based on these surface markers. So the key point is we're not trying to develop a technology to compete with this because they're way ahead of us. But there's a problem. The definition is based on a few surface markers of what a circulating tumour cell is. And as you all know, cancers are very heterogeneous, and that means the cells within the cancer are heterogeneous. And these cells are really rare. So we don't have a technology yet for looking at the heterogeneity in cancer cells. Why would that be important? Because that would relate to your treatment strategy. And so you can see that here. You don't need to know gene maps to understand that. So this is some primary cancer cells and some circulating tumour cells. And the key point is different colours. The colours relate to the level of expression of different genes. All you need to see, no one cell is the same. So that's the heterogeneity. So we wanted to develop a tool to allow us to look at the heterogeneity that was compatible with those counting devices, the fluidic devices, and we use magnetic nanoparticles again, of course. And so then we designed a surface where the cells could be captured, and we could look at that surface and just release one. So we could have the cells on the surface, we can see how they respond to drug treatment, and then we can choose to just release just one. And the way we do that is this is a semiconducting surface, so the same sort of surface used in all those physical devices and all the microelectronics industry. And it's got chemistry on it with antibodies, and we have it so that we need two stimuli to release the cells, light and potential, so voltage. So you can look at the surface with no voltage applied, nothing's released. You can decide, I want to release that cell here, this one is not responding to the drug treatment, or this one is responding differently, I need to understand more about that cell and release it. And rather than me tell you that, I'll show it to you. So here's 3 cells on the surface, here's a micropipette to collect them, you can see they won't come, you can watch this cell coil towards the pipette, and then recoil away. It's like, whew, that was tough. And then what we do is confine the light just to that one cell and apply potential, and we can come along and pick up that cell. And we can then clone that cell, analyse that cell, work out why it's different, work out what's different about it, see what treatments it responds to, and so perhaps have a better tailored treatment. So that brings me to the third technology I'm going to tell you about. You can see you can't pick up these 2 cells because they didn't have, my laser pointer's dying, I think. They didn't have the light applied to them. So this brings me to the last technology, and this relates to now taking those cells and working out how they respond. And so a cell biologist traditionally puts cells onto a surface, a flat surface. It's classic cell culture, and it's two-dimensional. And of course, cell biologists have recognized that most of us aren't two-dimensional. Some of us may be, but most of us aren't. And so they want to make balls of cells, little balls of cells, because they're a great mimic of actually a solid tumour. And so where these cells may be looked at on polystyrene or other plastic, your solid tumour in your body is not just sitting there by itself. It's wrapped in a whole series of proteins called the extracellular matrix. And so they really want to make these balls of cells and then immerse them into some sort of mimic or actually an extracellular matrix, a protein environment around cells, because that influences how cells respond. And then hopefully we can make better models of how cells will respond to drugs, because we know that from there to there we can get differences, and so limit the use of animals and when we go into people for drug trialing. And so what the problem is making these cells. So the way a typical cell biologist does it is she gets about 5 to 20,000 cells. She puts them into these dishes, which have got round bottoms. The cells don't like to stick to them, and waits for about 5 days, and the cells sort of move around. Cells like each other, so they form a bit of a party and start forming these balls. I quite like these 2 here. They can't work out whether to join. This guy says, you're a loser, I'm in there, and off he goes, and this guy gets missing out. So they make these balls of cells. They're really good at it, and they can do it quite effectively, but they have this one step. That's the ball of cells sitting in buffer or in cell media. Then they have to pick it up with the pipette and embed it in that extracellular matrix, a mimic of that protein that your cells are sitting inside your body. And there they lose maybe 40, 50%, so it's a problem. And you can see it takes 4 to 5 days. They can make quite a lot, but then they've got this tricky step. So we worked with this company called Scientia. They basically came to us right in the very beginning because the Australian Centre for NanoMedicine can do everything from the materials all the way through to the biology because of the people we have. And we designed a whole new 3D printer. And it's different to any other 3D printer. It's basically, you can see up here, this is all standard cell culture materials, so the biologist doesn't have to change their workflow. And then we have a series of pressure, you might say these pressure regulators, because we redesigned a head that spits out cells. And what's key about it is that we in my group designed the inks, the engineers in the company designed the head. We designed the inks, and the working together between the company and the academics in the same, or the bench researchers in the same labs all the time, came together, tuned the inks and the head so we get very high cellular viability, better than or as good as what you do in normal cell culture. But yet we can print very high densities of cells. And so that means we can then make these balls of cells very effectively. And so this is what the first printer looks like. So we design inks, so we have the cells with one part of the ink, we have a second part that cross-links, and you've got a chemist talking to you, so I had to give you chemistry at least once. So we design the inks of these extracellular matrix mimics, and we design them so they have things to adhere, the cells, maybe groups that you can cleave so that the cells can remodel them, and then the printer works. And so this is the cartoon of the printer showing, so the cell biologist does all their normal workflow, then the printer is actually in an incubator, so it's sterile, and then we print some ink with no cells to form the extracellular matrix around the cells, and then we print a ball of cells, some cells that go in there. Because we're much higher cell densities, we only have to wait a couple of days, and then you have your little ball of cells. And so we can then, and just to show you, this is what it really looks like. Here's the ink and here's the ball of cells. So this is the ink forming that cup. Here's the balls of cells, and after a few days, or even after 3 days, we have, actually after 2 days, we have one of these balls of cells called a spheroid. And so then just to show you that they look, so this is the ones the biologists would make. These are ours. The contrast is a little bit different, maybe because of the light, but basically they look the same as we go through the ball of cells, and then they respond to drugs the same. And so we can then do it for all sorts of other different assays. So infiltration assay, you may not be able to see this for, say, T-cell therapy. As you know, that's a new form of therapy for cancers. We can do migration assays. We can do all sorts of different things. It's pretty early days, but the first printers are being made now, the first ones that go to the beta testing instruments to research labs around the country. So it's designed for research initially. So I'm going to stop here, pretty much. I'm not going to go to the last bit, but I'm going to tell you that those technologies I talked to you about all link in together. So we have a diagnosis technology, something for looking at rare cells. It could be for diagnosis or prognosis. So we find out there's a cancer very early on. We look for secondaries. How do these cancers respond to drug treatments? We make 3D models of your solid tumour to see how it responds to drug treatment. And we've actually also designed sensors that go inside the cells to see how they respond to drugs. So with that, I just really want to thank the Chief Scientist again for inviting me. I'd like to thank you for taking that very valuable time this morning, that hour of sleep, and coming to hear me speak. Thank you.